Thank you for the opportunity to participate in this afternoon's session. So I've been charged with trying to discuss tissue handling and dissociation and perhaps the specific challenges that we might have as we generate a human cell atlas. So I'll not only cover the challenges, but also perhaps some potential solutions, or at least some work packages that are in place to generate those solutions. So in doing that, I'll call on some of my own or our own experiences locally, but also look to examples from others that are generating data for the HCA. So as we know, there are two major branches of the HCA, a spatial branch that basically takes intact tissue in order to preserve the anatomical relationship between cells. But this is relatively low throughput, and the cellular or nuclear branch is much more high throughput, so it's likely here that the data or more data will be generated. But to generate this data, we inevitably need to take tissues and dissociate them into single cell suspensions. And there are different sources for these tissues, as some of the previous speakers have touched upon. So we have tissues that come from live individuals. So these may be organs that are removed for cancer or disease. Tissue biopsies that are often done for cause, although as we've heard in the gut, you can, of course, do screening biopsies more readily. And then perhaps a really rich source of tissue for us as we build an atlas are organ donors that consent for research. So here, one of the potential advantages is that, of course, we can sample many organs from the same individual. And then there are deceased sources of tissues in terms of individuals undergoing rapid postmortems. So I would say that there's probably going to be no one solution in terms of a tissue source, and each of these have their advantages and disadvantages and practicalities or practical difficulties that we'll need to overcome. But what we're aiming to do in our efforts is obviously to take these tissues and to move to a point where we get single-cell suspensions, generates sequencing data that faithfully reflects both the cell diversity within that tissue, but also faithfully reflects as much as we can the cellular transcriptome actually in vivo. And so there are challenges, I think, that face us, particularly with regards to how we process our tissues. So the first is, how do we preserve the transcriptome as we move from A to B, so as we move from an organ or tissue biopsy to generating the data. And as I say, there are challenges that will be specific for our organ source, but for example, when considering organ donors, we know that some of these donors come to donation because of brainstem death. And so in these individuals, we can more or less retrieve tissues after ventilation is stopped, so artificial ventilation is stopped. But in contrast, we can also access deceased circulatory death donors. And in these, there's circulatory death and a stand-down period of time prior to the retrieval of organs, and so prior to us getting our research samples. So one question is, how might that warm ischemic time during circulatory decline affect the transcriptional landscape? As well as that, in the UK, and this can be also applied elsewhere, our organ donor network is national. So this means that potentially there may be an organ that becomes available, for example, in Scotland, in Glasgow, that we would want to analyse here in Cambridge. Therefore, there's the time taken for getting organs from one end of the country to the other. Now, traditionally, clinically, what we've done to preserve the state of those organs is to flush them and then transport them on ice. And we know that's effective in terms of just the basic pragmatic clinical fact that we transplant organs that have been stored for up to 24 hours and they still work in individuals afterwards. But how this affects the transcriptome of cells isn't very well characterized, and that's another area we need to address. So I would suggest that the second challenge that we face is how do we sample cells that have very tight associations with the connective tissue scaffolds and with adjacent cells. And that may differ in different tissues. So, for example, the brain may be more easy to dissociate than, say, the kidney or the muscle in particular, say, which has lots of or a very dense connective tissue network. And also the characteristics of the cells themselves, their size, or whether they have extensions that are very lengthy, are going to be challenging. And we not only need to develop protocols that may be specific for organs, but also specific for different parts of even the same organ. So for the organ that I'm interested in, the kidney, dissociating the cortex, the outer bit, is much, much easier than dissociating the pelvis, which is really fibrous. So again, we need to generate protocols that allow us to look at all areas of each organ. And then the third major challenge, I think, is how do we sample both common and rare cells? So if I take you again to the example of the kidney, so kidneys have these working units called the nephron, and blood will basically come in here into the capillary loop, gets squeezed out to form a filtrate, and then this filtrate travels through these tubules out to the collecting ducts, which coalesce in the pelvis. So a lot of the tissue is epithelial tissue, and if we think of proximal tubules, for example, those cells are very well represented. But there are many rarer subsets. For example, the podocytes, these are a specialist type of epithelial cell found within the glomerulus. They're much rarer, and they're also a funny shape. They wrap around and almost hug the glomerular capillary, so they're difficult to get out. As an immunologist, I'm interested in the immune cells that live in the kidney, and they, again, are rarer than the epithelial cells. So how do we get at both common and rare cell types? So I want to take you through a few examples. Obviously, in 15 minutes, I can't take you through every example, but a few examples of workflows that are trying to address these problems. So in terms of preserving the transcriptional landscape, so Mike Stubbington has been leading a study, and this has been taking organs that are harvested for us by Karosh Sayed Parsi, who's a transplant surgeon here locally in Cambridge, who set up the Cambridge Biorepository for Translational Medicine. And Mike has then been taking samples from DBD and DCD donors, looking at spleen, esophagus, and liver, and putting those samples in hypothermosol, and then putting them on ice, and then comparing biopsies taken at time 0, 12 hours, 24 hours, and 72 hours, and doing 10x single-cell RNA sequencing. And then Oliver Stiegel at EBI has been helping to analyse this data. But at the moment, this is preliminary, and the snapshot that we have is just from one sample, which is from a male spleen. So you can see at 72 hours, immediately, if you look at the cell clusters, there are some cells that are lost if you store these samples on ice over time. But encouragingly, if you look at the map reads, at 12 hours versus fresh samples, they look comparable. Now, there's only one sample, but this is potentially encouraging because it means we could then ship organs on ice from around the country here and obviously similarly elsewhere. Okay, so what about the issue of sampling cells, some of these cells that have very tight associations with the connective tissue scaffold? So we've been carrying out a study, and as Sarah mentioned, this is funded by CZI, where we've been systematically comparing different dissociation protocols. So a simple mechanical dissociation, where you use scissors and razor blades to cut up the tissue, and compared this with mechanical dissociation plus coagenase, an enzyme, or liberase. When we apply these enzymes, we obviously need to incubate the tissue at 37 degrees. So we've now done this with 3 kidneys, and Kevin and John have been leading on this. And you can see that in essence, this is representative of all 3 types, you can see we get many more live cells, both immune and non-immune, so the immune being CD45 positive, if we use the liberase. But I was really interested, and again, this shows the benefit of attending these meetings and efforts to collaborate, in a presentation by Michelle Monge from Stanford. And she's not interested in the kidney, she's interested in the brain, but she's been systematically comparing different types of liberase and had compared the standard one that we were using from Sigma with one from Worthington branded Stenzyme. And you can see that whether she's looking at glioblastoma multiforme samples, so on the far right, or cortical samples, that when she used the Stemzyme preparation, she gets an increased number of live cells. And so this is something we're now trying out on the kidney to see if this is applicable in our tissues. And again, Michelle shared this with the hope that, of course, others will be able to try it across different organs. Now, One of the disadvantages of using any enzyme dissociation, so although it's necessary to get cells out, unfortunately what we're doing, I told you, is we heat the cells back up to 37 degrees. And I emphasise that when we transport organs, we obviously try and keep them cool. So is there any way that we can dissociate samples without heating them up? And the answer is yes, there are, because there are clearly organisms that have enzymes that exist in very cold environments. And this particular bacteria, Bacillus licheniformis, which was actually isolated from the soil in Himalayan glaciers, has an enzyme, so that protease enzymes that work in the cold. And so Steve Potter from Cincinnati has been working to compare the use of this psychrophilic protease against a standard mixture of digestive enzymes that we might use. And he's using this for just 15 minutes and keeping the samples at 6 degrees. So I guess his first question was, well, do we now lose cells that are difficult to get out? And Steve is also interested in the kidney, but actually in the developing kidney. So this is data that he generated on mouse P1 kidneys. So you can see that podocytes, which are those cells that I mentioned that sort of hug on to the capillary cells, that you're able to get those out even with this very minimal digestive protocol. And what Steve also very nicely showed is that if you look at gene expression in the samples that have been digested at 37 degrees, by 15 minutes already more than 300 genes show an increase in expression compared with the samples that are processed in the cold. By 60 minutes, it's more than 500 genes. And many of these are stress response genes and include important transcription factors, for example, FOS and FOSB. So I've been emailing Steve, and he's obviously very enthusiastic about this, and definitely these data look interesting, and we want to try it out on kidney samples. But what he wanted to convey to the meeting was that he's now sourced this enzyme from Sigma. It's a lot cheaper that way, and in his hands it works. He's also used it to generate nuclei for sequencing, so it may be of interest to others who are using nuclear sequencing. But what he did point out was maybe on the downside that obviously this is a protease that isn't specific for ECM proteins. So if you're wanting to take your samples forward to look at surface protein expression, then maybe there are other ways we need to think about doing that. But I think encouraging. Okay, now another method of potentially dissociating without going through all the sort of manual disaggregation that I mentioned is actually using a microfluidic channel device. And Gerard Hown, also who was at the CZI meeting in January, presented this data. And basically what they've done is to produce this little device where you put cells in at one end and you force them through channels that are smaller and smaller to generate single-cell suspensions. And although I think at the moment this isn't practical, say for the average lab, that maybe in the future this may provide a more reproducible way of disaggregating samples. Okay, so what about the final issue, the sampling common and rare cells? Well, I guess there are three broad strategies. We can try and flow sort cells based on known markers, but this obviously wouldn't get us perhaps to the ionophore where we don't know the markers to start with. We can then try relative enrichment strategies, for example, using max speed separation, and we've been doing this for immune cells, so selecting based on just the expression of something like CD45. Or you can, in an even more pragmatic and simple way, apply an enrichment protocol based on a cell property, like its size or density. And again, this is something we've been trying to systematically compare, so using a 44% Perkol gradient to try and enrich for immune cells. And you can see that if we use that in combination with Liberase, then we get a really nice enrichment of immune cells, and we've shown that over 3 different kidney samples. Okay, so now to the bit I guess I wanted to show you, or I perhaps couldn't resist showing you just a little bit of data, because although the protocols are really important, we're obviously all doing it to get to some data. And so I'm interested in kidneys, and we've been working on trying to better understand particularly immune cells in the kidney. And we've taken samples, so nephrectomy specimens, or those from organ donors. And I work on the Cambridge Biomedical Campus, so my lab is here in the LMB, and the hospital's just here, and this is a 5 to 10 minute walk. depending on how much of a hurry you're in. So it means that we can get the sample from the operating theatre, say from a nephrectomy specimen, and very rapidly bring it to the lab for processing. And I think this has helped us lots. So it's meant that we can get from the point of having a sample through to getting those cells through the 10x in less than 2 hours. And hopefully this minimises artifact. And so what we've been able to identify is that there are a number of cell types in the kidneys, including immune cells. It's allowed us to take a census, really, of all the cells. And in the non-immune nephron, you can see we're able to pick up things like podocytes, those rear cells, but proximal tubular cells, intercalated cells, distal tubular cells. And you can see immediately from this t-SNE that the immune cells shown here in green are relatively overrepresented. So this immediately tells us, although we weren't selecting for them, that in our processing protocol, we are definitely losing some of the non-immune cells, and they may be more sensitive to the things that we do. If we then take the immune cells and cluster those, you can see an incredible diversity. So 14 different types of immune cells, including a couple of types of NK cells and diverse mononuclear phagocyte subsets. Using this data, because we're profiling non-immune and the immune nephron, we can also start to ask, how does the non-immune nephron, so how do the epithelial cells interact with the immune cells? And one obvious way is via their expression of transcription factors. So if we look, their expression of chemokines. So if we look at the non-immune nephrons, so glomerular, proximal tubule, distal tubule, and look at various different chemokines, we can see that some are more highly expressed in the proximal nephron and some are more highly expressed in the distal nephron. And by looking at the receptors that are found on the surface of immune cells, we can then predict which cells would end up where in the kidney. Okay, so to summarize, I think that our main challenges when we're thinking about processing are firstly stability of the transcriptome, dissociating tightly bound cells and sampling all cells, including rare cells. And there will be different problems depending on our tissue source. I think we're going to need organ and tissue-specific protocols, and actually these really routine, systematic comparisons are going to be essential, but not just for us to do them in our individual labs, but for us to share them either by publishing them or actually sharing them, for example, on protocols.io. And I think, then people, say, looking at the kidney can see what people who are looking at the lung are using and see the things that we might be able to apply. So I'd just like to acknowledge the people that were involved in the work that I showed you there on the kidney, so particularly people in my own lab, Ben and John, Kevin and Alex, and then Sarah Teichmann and Sam Bechati, who've been phenomenally supportive, and Sam amazingly enthusiastic in terms of sourcing the samples, particularly the tumour samples, which you heard a little bit about earlier. Thanks to Steve, Michelle and Mike for giving me the other data that I've showed you, and for the people that fund us. So thank you very much.
Hello. First, I just want to, vouch for Steve Potter's method. I think it actually works really well. It's sort of our standard method now for our sequencing. And it actually gives us a lot lower number of dot runs for our sequencing. So I think it's definitely quite good. The question I have, though, is there a quantitative assessment of how the transcriptional state change upon dissociating epithelial cells? Because epithelial cells, by their nature, are connected together, right? Compared to their in vivo transcriptional state.
Yeah. So you're saying in the dissociation process, right, inevitably, if we're taking cells away from their neighbours, we will change their transcriptome.
Right.
And I think that that's a very valid point, which is why we have to use combined approaches where if we're keeping tissues intact in the spatial branch, then we've got a kind of check on the data that we're generating by dissociating. Right. So I think we have to use the methodologies in parallel.
I'm just wondering, in the future, would there be like a standard set of genes that we can query spatially that would
Yeah,
tell
I mean,
us that? Can we come
potentially,
up with that?
eventually, once we get enough data and we could compare spatial versus dissociated, then yeah, we could potentially generate those sorts of gene lists, say.
Thank you.
And I think going back to the first thing, getting good protocols definitely saves doing sequencing that may not generate, I guess, as I said, what we're aiming for is a faithful representation of the transcriptome. And although it's great that we get on and generate data for our projects, you kind of don't want to run before you can walk.
Wolfgang from Munich. Have you tried to do bulk RNA sequencing on the undissociated tissue and then on the suspension and compare that, how that differs? Because that would be another possibility to see how actually the dissociation whole procedure would affect it, right?
Yeah, so do you mean like bulk sequencing or?
Yeah, I mean bulk RNA sequencing from the tissue, that would be the truth, right?
Yeah.
And then bulk from your dissociated samples, that would be the comparison then.
Yeah, so we haven't tried that comparison directly. We certainly, for all of our kidney data, have taken samples for bulk RNA sequencing simultaneously, but that's a really good suggestion. And again, something that would be helpful to the community if we can make that data available, exactly what the changes are that happen during association.
Yeah, just related to that last question, I'm particularly interested in, are you getting distal loop epithelial cells out? And I would expect they're going to be hard. And
yeah, so as I showed you, the immune cells are overrepresented, but we can get distal tubular cells. We can get intercalated type A and B and principal cells out. So we are getting them, and I know, so chatting to a number of people who are working on kidneys, including Ben Humphreys, that they've had problems with fresh processing of whole cells like this in getting data on tubular cells. They seem to be particularly prone to dying in the...
Proximal's easy, it's the distal stuff that's hard.
Particularly, whether that's because they're phenomenally metabolically active, because they're, pumping stuff in a high energy way all the time. We see a higher mitochondrial gene content in the proximal tubular cells that we have.
Can I just ask about the immune repertoire? How variable is that? I mean, same for the GI and the lung, everything. We're just going to see this incredible immune diversity in adult organs. But what's your sense of the individual variation level of different sub-differentiated types or subtypes?
Yeah, so I mean, we've sampled 10 kidneys here. We can see some inter-individual variation, but we do see contributions to most of the cell clusters from most of the individual samples we see.
Good. Okay, if there are no more questions, let's thank Manava for a round of applause.